Yes, good afternoon, classy. I welcome you again to Metric Hub. Today I would like to present to you the chapter or the concept of cloning. I'm going to be talking about cloning today. Uh, just to begin with, I would just like to define what is cloning. I'm sure that you have seen question papers or you've seen different content which has been presented in trying to explain the concept of cloning. Cloning, I'm sure again we have seen the development of the footy cow. It is a typical example that has been used in explanation or in illustration of cloning, where a footy cow was produced through the process of cloning through which is actually through reproductive cloning. So let me just first of all give the definition of cloning. Cloning, we are saying, actually I'm saying cloning is making one or more genetically identical copies of an individual cells or organisms, where a number of, we are saying identical cells can be made one or more identical genetically identical copies of a cell or organisms that can be produced. That is called cloning. So what is happening is that we are saying this process of cloning, it has been just brought up as a result of genetic engineering process. So I have came up with a question or some questions that are actually like there to help us in terms of trying to comprehensively cover the various questions that might arise, the technical question that might arise. You can see on the board that I've illustrated there, uh, it is the cloning of a champion horse, which is being used in regards to this question. So basically, let me just give you and write the definition of cloning as I've said in the first place there. What is cloning? What is cloning? So I've said cloning, cloning, it is making, it is cloning, it is making, it is making, if you can see, we are saying we are making one or more genetically identical copies of individual cells, making it is making of one or more genetically identical copies, identical copies. We are making one or more genetically identical copies of individual cells or organisms, copies of individual cells or organisms. So in other words, we are saying there is one copy of a cell, or we have got a copy, we have got an organism that can be used as a champion, or that has got the desired qualities, desired characteristics, that is therefore used in the production of other identical, or it is used to produce identical copies that are a replica of itself. So in that regard, we are saying that is cloning. So it can also be done through, it is a process that can be done also through injecting genes directly into a fertilized egg cell, whereby to say desired genes can be can be injected directly into an egg, into a fertilized egg cell. In this case, we are saying genes can be cut from the DNA of species. Genes can be cut from the DNA of one species and injected directly into the fertilized egg or eggs of another animal species. As the animal develops, as it develops from an embryo, the genes or the new gene is present in every cell of the body of that animal that it has been injected in. That is whereby to say it is injected directly into what? Into a fertilized egg cell. And I've said again, this process is done through genetic engineering. So I would want us to again define what is genetic. What is genetic engineering? Genetic engineering, this is basically defined as, or it is basically defined as, it is the manipulation, or alteration of the genetic of the genetic material of an organism, genetic material of an organism the genetic material of an organism is manipulated or it is altered, which we are saying it is, why is it done? Why are we manipulating, or why are we altering, or why are we changing the genetic material of an organism, which is actually to produce better or a new product? The reason for the manipulation or manipulation or alteration of a genetic material of an organism, we are saying to produce the desire to produce better or new product. When the genetic material of an organism is manipulated or it is altered, we are saying the intention of the scientist is to improve the organism or to get it improved in some way, which can also be as a way, or also to say there is a new product that is totally being developed. So as you can see, we have been saying cloning and genetic engineering. Cloning, actually, it is a process that is done under the concept of genetic engineering, where organisms can be developed by people. People have developed organisms, or they came up with the organisms that fit the interests or that fit the best qualities that they have desired, which is also called artificial selection, whereby we say qualities that are desired. Of course, in other natural terms, we can say when artificial selection is done, the organisms can be allowed to mate on their own, but there is just a control which is given to say which ones would mate. But in this case, we are saying there is a DNA component of an organism that is cut. It can be injected in a fertilized egg cell such that it can also grow, or it can also multiply as the organism is growing. Therefore, it is found in every cell of the animal or of the organism which it has been injected in. So without wasting much time, I'm going to dwell much in terms of explanation of this process of genetic engineering after we have understood, or after we have seen, after we have answered these questions that are here. The questions that are here, we basically just need to answer them. So therefore, we can be able to further understand what are we discussing and what is it that we are talking about. So you can see here the diagram below shows genetic engineering process. A donor cell was taken from the muscle cell of a male champion horse that was a stallion to create a new offspring. So in other words, I'm saying whenever you are given a question, or when you are given a scenario where there is a donor and a champion individuals, or champion and donor organisms involved, before you even look into many other areas that would arise, just get it right that you are talking, or we are being guided to understand the process of what? The process of cloning. The process of cloning is the one that would need to be taken into account in that case. So as you can see here, I'm going to use a green pen just for the explanation of our diagram here, which we can say, I can basically say this horse is horse 1, and then we have got also horse 2, which is this one, horse 2 and 1, the one on top there. As you can see, what is happening there is that we have got a cell from muscle tissue of a male champion horse, that is a stallion, which is being extracted. And then we have got also an ovum or an egg cell from a female horse, which is a male, that is shown there, which is horse 2. It gave the horse, it gave the ovum there. That is being shown here in this regard, in this scenario of B there. So what is happening is that an egg cell is taken from the what? From the donor. So I want you to understand to say, we can say, this one, we can give it as to say, uh, she can be taken as the surrogate individual, and then this one, we can define it as the donor. The donor is the one that is giving the DNA component, which is given a tissue cell. It is a donor. One, we can give it as a donor, and number 2, we can give it as a what? As a surrogate. So what I'm saying is, take note of these terms and take note of these variations. So we are saying the surrogate is giving an egg cell, which is going to be removed. Its nucleus, the nucleus of the egg cell is removed and discarded. So in other words, we are saying this nucleus here, this nucleus, it is being removed. It is being what? It is being removed. As you can see there, that is why you see we have got an empty an ovum without the nucleus yet, because the nucleus has been thrown away, which is reason because we said that the nucleus is not wanted because the surrogate does not contain the desired characteristics. And then we have got the muscle cell from the champion. One thing that I want you to understand is that the champion, it is always having the desired qualities. The champion always have got the desired, it always have the desired qualities. That is one thing that you need to understand. To say champion always have the desired qualities, and then the surrogate has got what? Unwanted or undesired. We can say undesired are therefore the ones that are being provided by the what? By the surrogate. It has got unwanted. It has got unwanted, undesired qualities, undesired or unwanted qualities. One thing that I want you to take note of, to say, as long as it is unwanted, therefore the nucleus must be removed. So therefore we get rid of its genetic information. And then, as you can see here, the nucleus is removed from the muscle cell, and then the nucleus cell is placed in the ovum of the male, and electric shock results in the ovum starting to divide. What is happening there is that scientists, after they cut after they take the nucleus from the muscle cell, it is stimulated through an electric shock, such that so therefore it must start to divide. It is just done to activate the process of what? Of division, which is what we call mitotic cell division, that is taking place there. So therefore it can grow. That is one concept that I want you to take note of. So we are saying that nucleus is the one that is being placed into the what? Into the ovum that was removed, its nucleus from the surrogate mother there. And then as you can see at C here, as you can see, we are saying the ovum divides normally, and then the embryo placed in the what? In the uterus of an adult female horse, which we can as well just refer to say, we can define as to say the adult female horse is the what one, which we can say it was the surrogate. It was just a surrogate, which is just going to be used as what? This one is just used as what? It is used, it is being used as an incubator. It is just used as an incubator. It is just used for the development of the what? Of the embryo. Is used as an incubator. I'm sure you understand to say it is an incubator because it is only giving the conditions that would allow the embryo to develop until the gestation period is enough, so therefore it can be birthed or it can be what? It can be born. So the incubator is just it is just giving the conditions, the environmental conditions that are necessary for the development of the embryo after it has been placed into its uterus. It has nothing to do with the characteristics. The characteristics of this embryo that is being to be cloned or that is to be produced, it has got all the qualities of the what? Of the donor, of the donor or of the champion horse that we have named as one there. So we have got the embryo develops normally, and then we have got an offspring. Before we go to our questions, please take note that the qualities, the characteristics of this one, this offspring, it has got the characteristics, it has got the qualities of what? All the qualities are for the contains uh, uh, qualities of the what? Of the champion horse. Of the champion horse. So the characteristics that are there, all the characteristics that are in this new baby or into this offspring, it has got all the characteristics that are a replica or that are being from the what? From the champion horse there. Number, which is what? Which is horse one. Like I've said in the first place, horse one, it has got all the qualities that are being to be produced in the what? In the offspring that we are seeing here, therefore being born. So I'm sure you've heard and you understand this process of what? This process of cloning. How is it being done? Let us quickly answer the question that are below there. Question 3.5.1. Question 3.5.1. 3.5.1. We are saying the question says, name the genetic engineering process shown in the diagram above. We are say, I've said that the process taking place there is what? Is cloning. The genetic process taking place is cloning. I'm sure you can see, as I said, there is a champion horse and there is what? There is a surrogate horse. So it is called cloning. That is cloning. Question B, process that produced ovum B. What is the process? This is A, and then B. What process produced ovum B? Remember, we are talking about a process that has resulted in the formation of the egg there. So it is an ovum. The process that is involved there is supposed to be the one that you should understand to say it is oogenesis. Oogenesis, which is the production of egg cells, the production of ovums from a female or in the female organisms. We call that oogenesis, or else we can either also say that it is what? It can also be called gametogenesis. It can also be called gametogenesis. Gametogenesis because it is a process of gamete formation. It is gamete formation, hence we can say it is oogenesis or gamete formation because they are gametes that is being formed in B here. We are talking about the gamete that is formed in there, an ovum, which is produced. It is formed through oogenesis, or it can also be called gametogenesis because there's a gamete that is being formed there. Question 3.5.2. 3.5.2. 3.5.2. Why is the donor cell extracted from the champion horse? The donor cell, we are saying, why is the muscle cell extracted from the champion horse? The champion horse, because it contains the genetic information or genetic material that is required. The answer is saying 3.5.2 because it contains the desired characteristics. It contains the desired characteristics because it contains the desired characteristics, the desired qualities. We are saying, why is the donor cell extracted from the champion horse? The champion horse is the one that contains the desired characteristics that are to be produced. Therefore, we want to produce an individual, an offspring that is a replica of one of the champion. The champion contains the desired qualities. That is one thing that we need to understand. Question number 3.5.3, 3.5.3, 3.5.3. Explain why only the nucleus of the donor cell is used. Explain why only the nucleus of the donor cell is used. We are again saying that the nucleus is the one that contains the genetic information, the nucleus of the donor cell. The nucleus contains all the genetic information, of the what? Of the champion horse. Of the champion horse. I'm sure you can hear and you can see what we are saying. The nucleus contains all the genetic information of the champion horse, which is also desired. Only why the nucleus, we need the nucleus because it's the one that has got the qualities or the genetic information that is needed there. And then we go to the next question. The next question is 3.5.4. 3.5.4 is saying 3.5, a somatic cell of a horse contains 64 chromosomes. How many chromosomes would therefore be in structure A, B, and muscle cell D? But what I want you to understand is that a somatic cell, what is a somatic cell in the first place? I want you to understand what is a somatic cell. What do we mean by a somatic cell? It is all body cells except sex cells. That is a somatic cell. A somatic cell here, we are saying all body cells except sex cells. That is what you call all body cells except sex cells is what you call a somatic cell. A somatic cell. So we are saying it contains 64 chromosomes. It contains 64 chromosomes. And then how many chromosomes would therefore be in structure A? How many chromosomes will be in structure A? How many chromosomes will be in structure A? Structure A, how many chromosomes would be there? A, as you can see, we are saying this is a cell from the muscle cell. This is a cell from the muscle tissue of a horse, male champion horse. So we are saying it is a somatic cell. Therefore, nucleus removed and what? So from the muscle cell. So A, the number of here, it must have what? 64. It is supposed to have, actually it contains 64 chromosomes because it is a duplet cell. A, number 3.5.4, 3.5.4A, the number of chromosomes that are there, it is 64. As you can see, we are saying 64 because it is a duplet cell. That is A there. And then B, how many chromosomes are in B? In B, as you can see here, this is an ovum. Therefore, it has undergone meiosis. Number B, how many chromosomes are there? It contains half the number of chromosomes. which is supposed to be saying, or which is that to say it is 32 chromosomes because it has got half the number of chromosomes. B, it's an egg cell or an ovum that has been formed through oogenesis that we have talked about there, or which was formed through gametogenesis there. Hence, it contains half of 64. After meiosis, it contains 32 chromosomes. And then a mass cell, how many chromosomes would be there in C? In C, how many chromosomes will be there in C? C must contain must contain how many chromosomes? C, which is muscle cell in organism D. It is a muscle cell. Again, it must have the full set of chromosomes. We are saying it is 64 chromosomes because it is diploid. So number A and C, they are diploid cells. They contain all the number of chromosomes that are there. They contain the full set. Only question B, it contains half because it has undergone it has undergone what? The process of meiosis there. Explain why the ovum C cannot be considered a gamete. Number questions 3.5.5. 3.5.5. Explain why the ovum labeled C cannot be called a gamete. Labeled C. Why should we say that it is not a gamete? Where is C? C, as you can see here, it is not we are saying nucleus. It is the nucleus of the champion horse that has been produced. That has been placed in this egg cell. So C is not a gamete. A gamete cannot be diploid. This is a diploid cell. As you can see, it is diploid there. So what is the reason why it cannot be called a gamete? It is not a gamete because we are saying, or I'm saying that it is diploid, of which gamete is what is haploid. That is the reason why it cannot be called a gamete. It is it is a diploid. It is a diploid cell of which a gamete must be what? Must be haploid. A gamete must be haploid. I'm sure you can understand with me that a gamete is haploid. This, in other words, we can just say it contains the full set of chromosomes. It contains the full set of chromosomes instead of what? Instead of having only half. Instead of having only half of the chromosomes. So it is containing the full set, this one, C. It cannot be a gamete because it is having the full set of chromosomes instead of having only half. So it is diploid cell, of which a gamete must be what? Must be haploid, is one condition that we have said. We can also put it in different terms or in different phrasing, which we are saying it contains the full set of chromosomes instead of having only half, which is why it cannot be called what? It cannot be considered a gamete. 3.5.5 cannot be called a gamete because of that. So I can as well be able to give you other relative questions that might also come in this case. There are certain questions that would also come with regards to cloning, whereby you will be asked, for example, to say, why using a muscle cell instead of a sperm cell? Why scientists must use? Explain why. Explain why scientists must use or have used a muscle cell instead of a sperm cell. Instead of sperm cell of the champion horse. The sperm cell of the champion, the sperm cell of the champion horse. Why should we use that one instead of this? Why should they use why must they use the muscle cell instead of what? Instead of the sperm cell? So the answer in this case is that we must understand that the scientists can use the muscle cell because it is also diploid. A sperm cell is haploid. That is the thing to say. The muscle cell is what is diploid, or it has got the full set of chromosomes. Whereas whereas a sperm cell is what is the sperm cell is what is haploid. That is one reason that can be given there to say explain why scientists must use muscle cells instead of sperm cell of the champion horse. It is because of the champion horse. It is because we are saying the muscle cell is diploid. It contains a full set of chromosomes rather than half when it is a sperm cell. The other question is advantages of cloning. What are the advantages of cloning? Advantages, it must also be taken into account, this one. Advantages of cloning. Advantages of why is it important to do cloning? The advantages of cloning, what are these? One, we are saying to produce organisms with desired traits or desired characteristics. One, cloning, we are saying it helps to produce organisms with desired traits. The traits that can be, we are saying, e.g. appearance, that is, for example, e.g. in terms of appearance, the good traits, it can be in terms of appearance, it can be in terms of health, it can be in terms of the yields. As for example, we are saying yields, like for example, in the production of the dairy cows, the food was produced because it managed to produce a lot of milk, other than the ordinary dairy cows that were, the ordinary cows that were there. It can also be for advantages of the shelf life in terms of agricultural produce. Agricultural produce, we need them to stay longer in the shelves. So if they are cloned, we can as well be able to have them for a longer time. Another point is that why is it important to do cloning is that it also helps in the conservation of threatened species, protection of threatened species, production of threatened species. What is happening is that the endangered species, given that they might be coming to extinction, we can as well be able to produce many of them, having just one or two of them that are still available or that are still alive. So endangered species can also be protected or can be what can be preserved, protection or preservation of, which we can say preservation, I think can be the best word. Preservation of threatened species can be fine. And then we are saying to create tissues or organs for transplant. Cloning help also, cloning, it helps also creation of tissues. It helps in creation of tissues, creation of tissues or organs for transplant. Tissues or organs for transplant. When certain problems or diseases or health conditions that might be complicated, through cloning, we are able to create tissues that are needed for certain transplant. Therefore, it is an advantage. It is an advantage, as we can see there to say advantages of cloning. They are not asked in this question or in the question that we've been doing now, but I've just managed to say, let me put them through for the class to take note of them and to understand what is happening. Still on the same question of cloning, sometimes they might also ask you to say, why is it that the nucleus of the surrogate of, or the nucleus of the surrogate mother who has given the ovum was removed? The ovum's nucleus was removed because we wanted to get rid, or the scientists wanted to get rid of the characteristics of the mother, of the surrogate mother, just to remove or to get rid of the characteristics of the surrogate mother. That is why the nucleus was removed from the what, from the ovum there. As you can see, having said this, I would like us to just complete this unity by looking at some other party questions that might also need to be understood. What is reproductive cloning, therapeutics cloning, which I just wanted you to, I wish that you can as well be able to understand their terms or their meanings there. What is reproductive cloning? Reproductive cloning, like I've been saying in the beginning, we are saying it is the transfer of genetic material of an adult cell to an egg cell where the genetic information has been removed to produce a cloned animal. In this case, I'm sure you have seen that reproductive cloning is similar to the scenario that we've been discussing right now, which we are saying reproductive cloning, the genetic material, it is transfer of genetic information, or let me just say information. It can genetic information or material, genetic material or information of an adult cell of an adult cell. An adult cell, it is whereby to say of an adult cell to an egg cell where the genetic information has been removed. Transfer of genetic information or material of an adult cell that I want you to see, as we have discussed above there, to say you can see that the champion horse muscle cell was taken, and then it was put into an egg cell whose nucleus was removed. So therefore, an adult cell to an egg where the egg cell nucleus was removed to an egg cell whose nucleus is removed. So reproductive cloning, it is actually illustrated through the diagram or the board information that I have displayed there, where we have got the champion horse muscle cell being taken, and then it is put into the what? Into the egg cell of that other mother that we said it is the surrogate mother, which we of course again said that the nucleus of that egg cell has been removed. So then we can put the nucleus of the muscle cell of the champion horse, and then define therapeutic cloning. It is therapeutic cloning. This one, therapeutic cloning is the use of embryonic or adult stem cells to produce new tissue cells to treat diseases. The use of embryonic or adult stem cells to produce new tissue cells for the treatment of various diseases. Use of embryonic it can be embryonic or adult. It can be embryonic or adult stem cells, which I defined that, I'm sure that I defined what is stem cells. Use of embryonic or adult stem cells, adult stem cells to produce new tissue cells to treat diseases, to produce, which we are saying to produce new tissues to treat diseases, to produce new tissues to treat diseases, to treat diseases. I'm sure you understand to say the therapeutic cloning. We are saying embryonic or adult stem cells are used or are produced are used to produce new tissue cells that can be used to treat diseases. And then stem cells, I already defined what is stem cells. We said all body cells except what? Except except sex cells. I have already defined this one. It is already defined. We defined it there upstairs. At the beginning there. And then what is embryonic? What is embryonic stem cells? Embryonic stem cells, these are stem cells in the embryo that produce new cells that begin to differentiate into different cells or different tissues. I'm sure you can see we are saying stem cells in the embryo. These are stem cells in the embryo. Stem cells in the embryo that produce new cells. In the embryo that produce new cells. That produce new cells. As you can see, stem cells in the embryo that produce new cells that begin to differentiate into different cells. New cells that differentiate into new cells. New cells that differentiate into new cells. That differentiate into new cells. That's what we call what? That's what we call the embryonic cells, stem cells in the embryo, or that are in the very young, developing young ones that produce new cells that differentiate into new cells. Adult stem cells, the stem cells in the mature tissues in an organism that produce new cells, that is adult stem cells. We are saying stem cells in the mature tissues in an organism that produces new cells. And then the last but not least is gene therapy. Gene therapy, we need also to understand to say it's the introduction of genes into an affected individual for therapeutic purposes. Gene therapy We have talked about treatment of diseases through cloning. So it is gene therapy. We are saying it is the introduction it is the introduction of new genes. It is the production of new genes. Production of new genes. The new genes into an affected individual for therapeutic purposes. New genes into an affected individual for therapeutic purposes. Individual for therapeutic purposes. So, introduction of new genes into an affected individual for therapeutic purposes is what we call gene therapy. Basically, what we understand, I mean, you need to understand that there are certain diseases that might affect organisms. So, scientists would use the new genes that might be used be used to replace those difficult or those affected cells, such that they can be able to develop and multiply. So in that regard, we are seeing gene therapy. It is actually the introduction of genes into an affected individual for therapeutic purposes. Therefore, the organism can start to function properly. So in this regard, I'm sure that cloning, we have tried to look at it in different areas or different angles that it can be asked. So thank you very much for your time, and I hope that the class or the lesson of today is going to actually give you a good understanding. It's going also to give you a good reference for studying, such that you can be able to pass our exams that are coming soon. All the best, and God bless you. Don't forget to subscribe, so therefore you can be able to see our next presentations, our next postings, our next classes. They are going to be posted as regular as possible in order for the class to keep updated. All the best, and tell a friend to tell another friend that there is Metric Hub, which have been brought to our platforms to actually simplify and make metric passing easier. We explain and we go through all the articles, all the different scientific terms, and break it down, dissect them, so therefore we can be able to understand why the question is giving us that as an answer, or why do we take that as an answer. Thank you so much. Don't forget to subscribe, and good day.